Assalamu alaikum, dear students. I am Muhammad Rauf. Welcome to the course Principle in Animal Life 2. This is lecture number 15. The topic is Eukaryotic chromosome. Discussing agenda for the topic is Introduction to Eukaryotic chromosome, Physical structure of chromosome, Chemical composition of chromosome, Organization of a eukaryotic chromosome, chromatin, autosomes and sex chromosomes, sex-influenced traits, sex-limited traits. Lecture outcome. After watching and listening this lecture, students will know about eukaryotic chromosome and its various aspects. Introduction to eukaryotic chromosome. A cell's DNA, its genetic information is called its genome. Genome is the set of all the genes in a cell or an organism. The overall length of DNA in a eukaryotic cell is enormous. Eukaryotic DNA consists of coding and non-coding regions. So because of the presence of the non-coding regions, their length becomes much greater as compared to prokaryotes. Here on the right side of your picture, you can see a eukaryotic cell's nucleus where the chromosomes are in condensed form. If you take out all the chromosomes from a single human cell and unwind their DNA and then join their DNA end to end with each other, then the overall length of that DNA strand will be up to 2 meters. Means the DNA inside your single body cell has an overall length greater than your height. In another comparison, we compare the size of the nucleus with the length of the DNA. So eukaryotic nuclei are usually 5 to 10 micrometer in diameter, and the DNA content is 250,000 times greater than this value. So just imagine an area of 5 to 10 micrometer diameter is housing a molecule which is 250,000 times greater than its diameter. Before the cell can divide to form genetically identical daughter cells, all of its DNA must be copied or replicated, and then the two copies must be separated so that each daughter cell ends up with a complete genome. The DNA is in association with proteins in the interphase, but as the cell commits to divide and form daughter cells, then this diffused form is condensed several times and discrete units are formed which are called chromosomes. So as the cell, the somatic cell divides into daughter cells, it is important that the daughter cells receive the exact amount of DNA as that of the parent cell. So therefore, the process occurs in a highly sophisticated way. We are talking about mitosis. Now, the cell replicates its genetic material because the cell is committed to undergo division and the daughter cells will be formed. So the DNA must be duplicated so that each daughter cell can receive the exact amount of genetic material. So what we basically mean to say here is that DNA is in association with proteins and exists in a highly diffused form, means there is a loose connection between the DNA and associated proteins in the interphase. But as the cell commits to divide, then this association between the DNA and the associated proteins is strengthened and the DNA undergoes several degrees of condensation, and as a result discrete structures are formed which are called chromosomes. The chromosome word comes from Greek. Chroma means color and soma means body, so named because they take up certain dyes used in microscopy. Chromosomes are invisible in their natural form, and the geneticists or the cytologists uses a technique called staining in order to make their studies more detailed. So after the staining procedure, the chromosomes takes up the dye, therefore they are called chromosomes. Each eukaryotic chromosome consists of very long linear DNA molecule associated with proteins. The DNA molecule carries several hundred to a few thousand genes. Now, genes are the functional units of DNA. Genes control different traits. They are responsible for the synthesis of enzymes, for the synthesis of proteins, and some genes regulate other genes. So the number of genes in an organism is a characteristic of each organism of the species. For example, the humans have 25,000 genes, bacteria have 3,000 genes. So it is something which is related with the structural complexity of the organism. Now, physical structure of chromosome. The physical structure of chromosome is here on the right side of your picture. You can see there are three important parts. First is origin of replication. Origin of replication is a site where the replication of the chromosome starts. The gene, the DNA also has their origin of replication, and the chromosome has their own origin of replication where their replication begins. Second is centromere. Centromere is a disk of proteins. Centromere is composed of DNA, and this centromeric region houses a structure called kinetochore. Kinetochore is a disk of proteins which is located on the centromere, and this kinetochore provides the site of attachment for spindle fibers during cell division. The third important region is the ends of the chromosomes. The ends of the chromosomes are called telomeres, and they are made up of DNA sequences, and certain sequences are repeated thousands of times in their telomeric regions. For example, in the human DNA, the telomeric region consists of thousands of repeats of specific sequences T, T, A, G, G, G. Now we come to the chemical structure of chromosome. So the chromosome is composed of DNA and associated proteins. The associated proteins can be histone proteins or non-histone proteins. The histone proteins are further classified into various types: H2A, H2B, H3, H4, and H5. Non-histone proteins can be different types of enzymes used in replication or transcription, or different types of proteins that are used in converting the diffused form of DNA into compact structures called chromosomes. Now, heterochromatin and euchromatin, even after interphase, the centromeres and telomeres of chromosomes, as well as other chromosomal regions in some cells, exist in highly condensed states similar to that seen in a metaphase chromosome. The type of interphase chromatin visible as irregular clumps with a light microscope is called heterochromatin. To distinguish it from the less compacted, more dispersed euchromatin, because of its compaction, heterochromatin DNA is largely inaccessible to the machinery in the cell responsible for transcribing the genetic information coded in DNA, a crucial early step in gene expression. In contrast to looser packing of euchromatin makes its DNA accessible to this machinery, so the genes present in euchromatin can be transcribed. The chromosome is a dynamic structure that is condensed, loosened, modified, and remodeled as necessary for various cell purposes, including mitosis, meiosis, and gene activity. So here we are discussing the two forms of chromatin. Chromatin is composed of DNA and associated proteins. So in the interphase, if you observe the chromatin with a microscope, you will see two distinct regions. Some of the regions will be darkly stained, whereas other regions will be lightly stained. Why? So, because the regions where the association between the DNA and associated proteins is stronger are present in a condensed form, and they are called heterochromatin, whereas other regions where the association between the DNA and associated protein is loose is called euchromatin. So heterochromatin is an area which is inactive, means it is impossible for the transcriptional machinery to access a gene which is present in a highly condensed area as compared to a gene which is present in less compacted area. So you can say that heterochromatin area either completely lack genes or the genes are present in very uh, minute number, whereas euchromatic regions are those regions where DNA or where the genes are in abundance, and they are gene-rich regions, and usually housekeeping genes are located there, which are transcribed on regular basis. So now the organization of a eukaryotic chromosome. Just as we said earlier that during interphase the DNA is present in diffused form. It has loosened connection with the associated proteins. Although the heterochromatic and euchromatic regions are still present there, but all the area is in a diffused form, and as the cell commits to divide, the association between its DNA and the associated proteins is strengthened, and there is several degrees of condensation, and at last a eukaryotic chromosome is formed. So the DNA of each human chromosome is thousands of times longer than the diameter of the nucleus. We have already discussed that all of this DNA can fit within the nucleus because of the multi-level system of packing, coiling, and folding. So there is several levels of packing, coiling, and folding, which enables a 2-meter long DNA molecule to assemble in an area which is 250,000 times smaller than its size. It is only possible through packing, coiling, and folding. A crucial aspect of DNA packing is the association of the DNA with small proteins called histone proteins. These are the histone proteins, and they form association with the DNA. So there are 5 types of histone proteins as discussed. The 4 types H2A, H2B, H3, and H4, they form the nucleosome. Nucleosome is the structure formed by the combination of DNA and histone octamer. Usually in a nucleosome there are 8 histones. The 4 histones, each of them exist in the form of a pair. So a total of 8 histones are wrapped around by DNA molecule. In fact, histone proteins account for about half the mass of eukaryotic chromosomes. Means they are so large in number that their amount is equivalent to half of the chromosome mass. This complex of roughly equal amounts of DNA and protein is called chromatin. So we have already discussed chromatin, which is the DNA plus associated proteins in a diffuse form. The unpacked double helical molecule of DNA has a diameter of 2 nanometer. So just imagine how much packing and coiling and folding takes place. This is a DNA molecule without association with histone proteins, and it has a diameter of 2 nanometer, and Look at here. The diameter of a duplicated chromosome, a condensed chromosome, is equal to 1400 nanometer. This is only one duplicated chromosome. There may be many more, just like humans have 46. So we are discussing here only a single chromosome. And you can see here, this is the first instance where the histones become associated with the DNA. Then there is another degree of coiling. So after the association of DNA with histones, a 10 nanometer, uh, you can say, a rope or fiber is formed, and then this 10 nanometer rope is further condensed via coiling, and it becomes 30 nanometer, then 300 nanometer, then 700, and finally 1400 nanometer. Let me tell you one thing, that this higher degree of chromatin folding is still a mystery, and still we don't know what type of process usually occurs, but these are, you can say, these are assumptions by the scientists, that the higher degree of folding and coiling may occur in this way. So, at the first level of packing, histones attach to the DNA helix to form a 10 nanometer fiber. Here, the DNA is associated with histones, and 10 nanometer fiber is formed, which is called the unit of chromosomal packing. The DNA histone complex has the appearance of beads on a string. You can see here, this is a DNA molecule and it is in association with histones. So you can say it is beads on a string. Each bead, called a nucleosome, consists of DNA twice around a protein core of 8 histone molecules. So nucleosome is the basic unit of chromatin remodeling, and the nucleosome consists of a histone octamer, and the DNA is wrapped twice around a histone octamer. In some books you may see 1.7 times, but here in that book they have mentioned that the DNA is coiled two times, twice around a histone octamer. Now, four types of histones form the nucleosome: H2A, H2B, H3, and H4. The fifth type, which we discussed a bit earlier, is called linker, because it is present between the two histones. It is present in somewhere in that region. That region is called linker DNA. So the fifth histone is present in that region, and therefore it is called a linker histone. At the next level of packing, the beaded string coils into a 30 nanometer fiber. So here DNA without histones is 2 nanometer. In association with histone, it becomes 10 nanometer. Then this strand is further coiled and becomes a 30 nanometer chromatin fiber. This 30 nanometer chromatin fiber coils further into a loop domain with a diameter of about 300 nanometer. You can see here it has further coiled and formed a 300 nanometer fiber. Successive levels of coiling and folding enables a huge amount of DNA to fit into a tiny nucleus, just like we discussed earlier that the eukaryotic nucleus is 5 to 10 micrometer and it houses a DNA molecule which is 250,000 times greater than its diameter. So this is because of several levels of coiling and twisting, which makes it possible. DNA packing tends to block gene expression by preventing RNA polymerase and other transcription factors from contacting the DNA. Now, if the DNA is in association with histone proteins, if it is wound around histone proteins, then the replication machinery or the transcriptional machinery cannot access such a DNA molecule. Higher levels of packing can therefore inactivate genes for long term. Just said earlier that if the degree of packing and condensation is greater, then such area is usually termed as heterochromatin, and the genes in that area are not transcribed. Genes with highly compacted chromatin such as the mitotic duplicated chromosome as well as varying compacted regions of interface chromosome are generally not expressed at all. So if there is greater degree of condensation, greater degree of coiling, then such a DNA strand cannot be accessible to the transcriptional or replication machinery and such area becomes inactive. It is usually a part of the gene regulation where when an organism needs the product of a particular gene, so that genes come from the condensed to a less condensed form. And when the organism does not require the product of a gene or it needs to shut up a gene, then it is changed from its loosely condensed form to highly condensed one. So here you can see that the condensation is 10,000 times. When a DNA molecule changes into compact structure called chromosome, it has to condense 10,000 times. Now we come to the number of chromosomes. A diploid cell consists of two N chromosomes where there are two sets of chromosomes. Each chromosome is present in a pair form. They consist of homologous chromosomes. Homologous means the chromosomes which have same centromere position, which have same bending patterns, which carry the same genes, although alleles may be different. So diploid number is the one where each chromosome is present twice. There are two copies of each of the chromosomes, and in haploid number there is only one copy, just like humans have 46 chromosomes, and their gametes, the sperm and ovum, has only 23 chromosomes. Similarly, the Drosophila melanogaster has 8 chromosomes. Corn plants have 20 chromosomes, and then their gametes have half number of chromosomes as that of their somatic cells. Now, important thing here is to note that the number of chromosomes in the nucleus of an organism is characteristic of a species. It is something which does not depend upon the size of the organism or the structural complexity of the organism. So, just an example, dog has 78 chromosomes. Humans have 46 chromosomes, and mouse has 40 chromosomes. So there are differences in their size, in their cognitive level, but the chromosome number does not suggest anything. Chromosome number does not depend upon the size or the structural complexity of the organism. Humans and other animal species have one pair of sex chromosomes that are not completely homologous. Now, there are 23 pairs of chromosomes in a diploid human cell. 22 are homologous pairs, and one of them is non-homologous. When we talk about the human female, human male, they have 22 pairs of autosomes and one pair of sex chromosomes. Autosomes are those chromosomes which carry out other functions. The genes located on them carry out other functions, and they have no role in determining the sex or characters related with sex. It is only determined by another type of chromosomes which are called sex chromosomes. So in the human male, the sex chromosomes are not homologous completely. One is X, the other is Y, so they are called non-homologous, whereas in the human female, even the sex chromosomes are homologous. Now, sex chromosomes and autosomes. Sex influence traits. Sex influence traits are expressed in both male and female, but expressed differently in each sex. Means these traits are present both in male and female, but they are expressed to varying degrees because of the sex. In addition, the inheritance of these traits can be dominant in one sex but recessive in the other. These traits are often controlled by autosomal genes and are related to the effect of hormonal differences on gene expression. Pattern baldness is an example of sex influence trait. This is expressed more often in males than in females. Now, pattern baldness is expressed differently, means the genes controlling this trait are expressed differently in the male and female. You can see here that the same genes are autosomal dominant in males and autosomal recessive in females. The same genes are behaving in two different ways in both sexes. In the human male, even if one copy, defective copy is present, then the human male will develop baldness. But A human female will only develop baldness if both the copies are defective. Now, this is something very interesting, that the uh, same gene located at a particular loci is behaving in two different ways in both sexes. The discrepancy is related to differences in testosterone level. The males have 20 times greater levels of testosterone than in females. So therefore, those genes are usually under the effect of hormones. In this case, the abnormal environment, hormonal environment, and the genotype interact to determine the phenotypic expression of the genes. The genes are also under the effect of the environment. You already know about the epigenetics, the nature and nurture debate. So the genes do carry a specific set of information, but sometimes that information changes with environment. Sex-limited traits. Sex-limited traits are inherited by both male and female, but are normally expressed only in one sex. Now, if we make a differentiation of sex-limited traits with sex-influenced traits. Sex-influenced traits are usually present both in male and female, but they are expressed to varying degrees in both sexes. And sex-limited traits are inherited both by male and female, but they are present only in one sex. One example is an autosomal dominant trait that controls precocious puberty. Means there is a trait which enables a person to attain puberty before the normal age. The gene is expressed in heterozygous males but not in heterozygous females. Now, such a gene, autosomal dominant genes, mean if one defective copy of that gene is present, it will express that trait, but it is behaving differently in males than in females. A male will develop this problem if only a single copy is present, but a female will not develop this problem if one copy is present. So this trait is dominant in males and recessive in females. Affected males undergo puberty at 4 years of age or earlier. Heterozygous females are unaffected but pass this trait on to half of their sons, making it hard to distinguish this trait from a X-linked genes. Genes that deal with traits such as breast development in females and facial hair in males are other examples of sex-limited genes, and are virtually all other genes that deal with secondary sexual characteristics. Now, this is a very important sentence. It means that the genes may be present in the human male. For example, the genes responsible for development of breast may be present in the human male. The genes responsible for facial hair may be present in the females, but they are not expressed there. Thank you very much. Allah Hafiz.